today video I'm going to talk about PCR machine and how it works. So firstly I will preview about the lab and how does it work. So come to the side. This is where the desk and everyone doing their research or writing their thing. This is where DNA extraction happens. There is the equipment, pipetting, centrifuging, and water bath, and all the bottles. All of them are usually sterilized. After doing the DNA extraction, we are going to do from the clean room to the DNA extraction. This is the laminar flow hood. It is under the UV light, so you have to be a little protective, and you have to wear gloves and lab coat to protect yourself and also the mixture. There's where we have to prepare the mixture of PCR, either by the original material of PCR. We have to prepare everything, or there is a kit you can buy online where everything, the mixture is already prepared. It's all connected so that there's no contamination or anything like that. So this is the PCR machine. In this experiment we are using the real-time machines. Everyone has to take turns using it, and it should be clean, as you can see there, so there's less contamination. So let's start by introducing what is PCR. We're going to take our samples and we are going to insert them in this way, in this pool, but it should be balanced so that no mixture, so that okay, so this is how it is when it's open. When it's closed, it can be like this. This is the closed stand. So for less contamination, when you are going to use it, you can open the lid like this. It's on and it's standby. This is where you should switch it off. Also, this is where you are going to visualize the results you get in the computer. Thank you so much.
Hi everyone, let me introduce you the polymerase chain reaction, or what we call PCR. PCR is a very useful technique when we are doing research work. So here is the small fragment of DNA. We will mix it with necessary reagents. Then we will put it into the microcentrifuge tube. And then mix. We will put it into the PCR machine. Then the machine will run by itself, and ta-ta! You got millions copies of this DNA. So, PCR is a laboratory method of amplifying gene until it reaches millions of billions of copies and to be detectable by other laboratory instruments. So now, let me explain the steps of doing PCR, the reagent part, the working principle and stage of PCR, and also types of PCR. We will start with the reagents. The first one is DNA fragment that we want to copy. This DNA is insufficient for instrument to detect. For example, we cannot perform gel electrophoresis because the DNA is too less. So, we need to amplify it to increase the DNA. Next, deoxyribose nucleotides, DNTPs. DNTPs contain all the nucleotides that require to synthesize new DNA in excess. The oligonucleotide primers. Oligonucleotide means small or short nucleotide. These primers will be the starting point to copy DNA. This primer can be designed by ourselves to suit our research purpose. And last but not least, the Taq polymerase. Taq polymerase is isolated from a bacteria named Thermus aquaticus. It functions as an enzyme to bind the free nucleotides onto the DNA template to synthesize new DNA. Usually, we will mix up everything, including the Taq polymerase, DNTPs, primers, MgCl2, buffer, and ultra pure water into one solution, and this solution is called master mix. Master mix is everything except DNA fragment. So let's talk about how the thing works in these tiny bites of the machine. The three basic phases of PCR including denaturation, annealing, and extension. These three phases will repeat, and repeat to form cycles. Before the cycle starts, we will perform initial denaturation step at 94 to 98 degrees Celsius for 1 to 3 minutes to ease up the denaturation step. After that, here comes the first step of the cycle, the denaturation step. This step is to break up the hydrogen bond in double-stranded DNA to produce single-stranded DNA by using high temperature. The temperature is 94 to 98 degrees Celsius. Time is around 10 seconds. Next step will be annealing step. The primers will search for the complementary sequence on the DNA template and bind onto it. This is to give a signal for Taq polymerase to bind onto it in the next step. Temperature is 55 to 65 degrees Celsius. Time is around 30 seconds. Finally, extension step. This is where the Taq polymerase bind onto the primer and call all the nucleotides to come nearer and then arrange until it produces a doctor-strand DNA. Temperature is 72 degrees Celsius. Time is according to the length of DNA fragment. Then, the DNA will go back to denaturation step, then annealing step, then extension step, then until 20 to 40 cycles. The final step is final extension. PCR is maintained at 72 degrees Celsius for 5 to 15 minutes to ensure all extension process complete. And that is all for PCR working principle. Let me introduce you some other types of PCR. The first one is real-time PCR. RT-PCR is the PCR that we can monitor the progress or the quantity of DNA throughout the PCR process. Moreover, there is also reverse transcriptase PCR. RT-PCR is used when we have RNA as template to produce cDNA. The third one is allele-specific PCR. In allele-specific PCR, the oligonucleotide primers is designed specifically to detect any mismatch on allele or specific gene. The fourth is touchdown PCR. The modification is at annealing step. Temperature is increased above the melting point of primer. Under this temperature, only specific complementary base will bind together. The temperature is decreasing for every subsequent cycle. This PCR aims to allow only specific sequence to bind. There are many more types of PCR in the market. Please do more reading on those PCR to get better understanding, because it will come up in our exam. That is all about PCR. We are from Group 5 and that is all about our presentation. Thank you.